Hi, I'm Satyajit Mayor from the National Center for Biological Sciences in Bangalore. Here to talk to you about membrane rafts as part of the iBio seminar series. I'm going to talk to you about membrane rafts in three parts. The first part is where I will talk about what are membrane rafts and our understanding, at least our colloquial understanding of membrane rafts. In the second part, I will try and describe our experiments that have led us to investigate what membrane rafts may look like in living cell membranes. And in the third part, I'd like to tell you what, in fact, our investigation in trying to understand these membrane rafts have revealed to us in terms of trying to understand the structure of cell membranes in a living motile system. Without further ado, let me move to the first part. What are membrane rafts? Well, the cell membrane is where membrane rafts are situated. The cell membrane, as you all know, separates the inside of the cell from the outside of the cell. And this barrier is what, in fact, allows the cell to transact its interactions with the outside world without having to worry about the outside world. And the cell is able to live within the membrane as a distinct entity. The cell membrane has a particular chemical composition. In fact, it is made up chiefly of lipids. There are about 500 types of lipids. There are several classes of these 500 types. These are defined by the head group of the lipids. The phosphatidylethanolamine is one type, the phosphatidylserine, the choline, and the sphingomyelin is another type of lipid. And they differ from the constituents of each class or each type of lipid differ from each other by the nature of the hydrophobic acyl chain that's attached to these lipids. The length and the degree of unsaturation characterize the differences between these different types of lipids. An extremely important class of lipids is cholesterol, because cholesterol in most eukaryotic cells is about 50% in terms of the total content of the phospholipids at the cell surface. So in terms of mole by mole fraction, cholesterol is often 50% of the total lipid composition of a cell membrane. It's an extremely unusual lipid because it has a planar structure. It is in fact makes lipids behave in a very funny way when associated with them. But lipids in general, the different types of lipids have two distinct types of abilities to pack together. First of all, they form bilayers. And in these bilayers, the saturated acyl chain containing lipids have a tendency to pack a little tighter. And the lipids that have an unsaturated chain, hydrocarbon chain, they tend to, in fact, pack not as well and as compact with each other. And cholesterol, on the other hand, is able to modify the properties of these different types of lipids in different directions. In fact, the roles that cholesterol plays in the structure and organization of the membrane and different lipids in the membrane is as yet quite contentious. We're not very clear as to what exactly cholesterol does in the membrane, and I think the whole field of membrane rafts is really a search to understand what types of roles cholesterol and what functions cholesterol might play in cell membranes. I think to understand the cell membrane structure, it's important to know the history of how the cell membrane structure was elucidated. There's an excellent review by Michael Edidin. It's a timeline that in which he describes the different discoveries which led to the final elucidation, as we know today, of the structure of the plasma membrane, where he begins with the discoveries from Agnes Pockels, where she takes oil droplets and puts them on water and makes a unimolecular layer of oil on water, to the discoveries from Langmuir, where he suggested, I think for the first time, that lipids might in fact form bilayers and be in the form of a bilayer which could cover the hydrophobic surfaces or hydrophobic parts of these lipids to make long bilayer, two-dimensional bilayer-like structures. The debate between in the cell biology literature was how do you put the proteins into this bilayer, because at that time it was already known that the cell membrane constituted not only of lipids but also of proteins. And there was a debate between people who believed that the proteins and lipids formed a trilamellar-like structure, and then there was others who believed that it was simply a bilayer where proteins and lipids in fact interdigitated between each other. But what I'd like to point out that the history of this field reveals that there was an incredibly intertwined relationship between concepts that were borrowed from studies on artificial membranes with experiments that were done on cell membranes. And I think together the understanding of the structure of the cell membrane was slowly elucidated. A major breakthrough occurred when I think Singer and Nicholson put together a fairly revolutionary model at that time, the fluid-mosaic model, which was derived from observations of electron microscopic analysis of arrangements of proteins on membranes, and studies, in fact, from some early studies from Michael Edidin's work, where they looked at the diffusion of proteins on cell membranes. These observations were in fact coupled with an understanding of how membrane proteins could be embedded in the bilayer. They could be either peripheral or integral membrane proteins. And I think understanding how proteins were embedded in the bilayer, coupled with these observations from electron microscopic analysis of protein arrangements, led Singer and Nicholson to propose that the cell membrane was in fact a sea of lipids in which proteins were embedded as objects. And in fact, they were floating in this sea of lipids, which were constituted of a bilayer-like structure. This model, I think, has impacted our understanding of cell membrane structure quite substantially, and I think it's worth understanding what the central tenets of this model are today. Well, the model states that the membrane is an oriented two-dimensional solution of amphipathic proteins or lipoproteins, and lipids are in instantaneous thermodynamic equilibrium in this system. So it's a model about a passive fluid in which lipids are in fact dissolved. The consequences of such a model are that unless otherwise acted upon, there is generally no long-range order in a mosaic membrane within this lipid matrix, and that the lipids of a functional cell membrane are in a fluid state rather than in a crystalline state. In fact, they are free to diffuse all over the place rather than be gathered and stuck in one location. Of course, this is in the absence of any external influence. So the lipids in the cell membrane in left to themselves, or the lipids in the cell membrane, behave as if they are in a passive thermodynamically equilibrated system. And it is in this context, I think, that we should try and understand what are membrane rafts and why did people feel the need to propose structures such as membrane rafts. In fact, today there are over 2,000 hits that you can pick up in the PubMed. When you look at, when you just type membrane rafts, you pick up about 2,500 citations for lipid rafts. If you look at other descriptors of these membrane domains, you may pick up more. They're proposed to be involved in diverse cellular processes: membrane trafficking, membrane sorting, signaling, cell migration, They're hypothesized to be involved in the nervous system, in immune function, in nutrient uptake, in cell cycle, virus budding and entry, pathogen biology, cell motility, what have you. And I think they're thought to be involved in almost every aspect of cellular function that involves membranes in a cell. And of course, and that affects diverse biological phenomena. So what is understood by the term membrane rafts? Membrane rafts, and here again, I think it's worth going to some sort of a summary or perspective by some of the pioneers in the field. Kai Siemens, in a very influential paper in Perspectives in Nature, proposed that lipid rafts are regions of the cell membrane where cholesterol and sphingolipids can come together and thereby segregate in the plane of the membrane and attract other components to come and concentrate in these regions. He also proposed that there could be proteins that could stabilize these segregated regions, proteins like caveolin, for example, which can associate with the underside of the cell membrane and stabilize these domains so that they could hold a special lipid composition. So there are three features of these types of domains. One is that they're expected to be cholesterol and sphingolipid enriched. A second feature that we understand in terms of a descriptor for lipid rafts is that they are constituted of membranes that are resistant to detergent extraction. And we'll spend a little time about this notion of detergent extraction in the subsequent minutes of this part. And a third idea about lipid rafts is that functions in the cell that require the presence of cholesterol are also thought to constitute functions that require membrane rafts. So I think each aspect of this sort of trilogy of sphingolipid and cholesterol enriched domains, detergent resistant membranes, and a cholesterol sensitive functional context have provided I would argue, a colloquial understanding of what membrane rafts are today. But the structures, I think, remain elusive and definitely controversial. And I think this seminar is about trying to bring to you some of those controversies and also bring to you a search for membrane rafts in living systems, which we will talk about in the parts 2 and 3 of this seminar. So today, there are many models for lipid rafts, besides the one proposed by Kai Siemens, that lipid rafts are constituted of little lipid or proteins that are shelled by a small boundary lipid around them. Another concept of membrane rafts is that there are many different types of domains in cell membranes, where in fact you have a large number of different types of aggregates of different types of composition maintained in some fashion by cellular membrane composition, particularly by the cell maintaining a particular lipid composition. So these are the different types of ideas that are circulating about what these lipid rafts might look like in cell membranes. But at this point, again, I think it's important to understand why did Kai Siemens and his co-workers feel the need to propose a notion for like the lipid rafts. And I think an understanding of that comes from, again, looking back at some of the historical experiments that led to the proposal of the raft hypothesis. And this comes from studies from looking at the membrane composition of the different domains of an epithelial cell. The epithelial cell is a polarized cell. It has an apical surface and it has a basolateral surface. And these two surfaces have distinct lipid compositions. Now, if the membrane of all cells are a well-mixed system completely at thermodynamic equilibrium, one wouldn't expect, unless there are physical barriers, to have a distinct composition at one surface versus the other. And of course, in epithelial cells, we know there are barriers. There are tight junctions. These little dark patches that I draw here are on both sides. These are tight junctions that separate the apical surface from the basolateral surface. But that means that one needs a mechanism of generating a lipid composition from internal sources, where molecules of different stripes can be delivered preferentially to one surface versus the other. And that's only possible, Kai Siemens and Van Meer, Gerrit Van Meer at that time argued, when you can segregate these components during the membrane trafficking process from the Golgi to the cell surface. If you can segregate components of one type from the other, deliver one set of components, for example, cholesterol and sphingolipid enriched components to one domain and not to the other, then automatically the cell is capable of generating this heterogeneous composition in the two surfaces of the cell. The other remarkable observation they made was obtained by studying the lipid composition of enveloped viruses. They found that in the basal viruses that budded from the basal surface of cells had a completely distinct lipid composition from those that budded from the apical surface. In fact, some of the viruses that bud from the apical surface are the hemagglutinin virus, and the VSVG, the VSV virus buds from the basal surface. These viruses have very different cholesterol and sphingolipid compositions. The HA or the hemagglutinin influenza virus has a cholesterol and sphingolipid enriched membrane, whereas the VSV virus is in fact rather depleted of these components. So together with these two ideas, Kai Siemens proposed that the membrane of cells must in fact be partitioned in some form so that they can segregate domains, and these domains can be dealt with differently inside the cell. Another major player in bringing about the Raft hypothesis was the GPI-anchored protein. So GPI-anchored proteins are proteins that are in fact made in the endoplasmic reticulum and attached to a preformed glycolipid, which then brings the GPI-anchored protein attached to a lipid anchor in the membrane of the endoplasmic reticulum, after which the GPI anchor is in fact delivered to the surface of a cell, where it behaves like a lipid anchored component. This component, in fact, when observed in epithelial cells again, showed up at the apical surface of these epithelial cells. And that again suggested that perhaps these GPI-anchored proteins are also associated with a specific domain in these epithelial cells during the trafficking process, and then they are delivered to the cell surface in these specific domains and excluded from being delivered to the basolateral surface. In fact, some of the early work from the trafficking of these GPI-anchored proteins contributed substantially to the Raft hypothesis, because during the trafficking process, Debbie Brown and Jack Rose in 1992, in fact, published, I think, one of the classic papers in this field, where they showed that during the trafficking of these GPI-anchored proteins, they were associated with detergent-resistant membranes. And it was the association with detergent-resistant membranes that began in the Golgi and then continued on till these proteins were delivered to the cell surface. At about the same time, Nigel Hooper and colleagues had also discovered that a wide variety of glycosphingolipids and GPI-anchored proteins were all insoluble in a cold Triton, which is a detergent, a cold Triton extraction procedure. And putting these two observations together of cold Triton insolubility and the association with this Triton insoluble domain during the trafficking process, Debbie Brown and Jack Rose proposed that perhaps these detergent-resistant membranes reflect the membrane domains that are involved in trafficking of these specialized lipid components to the cell surface. Coupled with that notion of the detergent-resistant membranes, there were studies in artificial membranes where it was observed that cholesterol-induced phase segregation of homogeneously mixed lipid components. Neutral sphingolipids were also found to be clustered in these artificial membranes. So putting all these observations together, along with the notion that that if you cross-link these lipid-anchored components at the cell surface, one could often activate signaling mechanisms inside the cell, and that these signaling mechanisms were activated by cross-linking of these lipid components with no obvious cytoplasmic connection. So the cross-linking of these components with no obvious cytoplasmic connection implied that there might be some transbilayer or transbilayer connection between the outside of the exoplasmic leaflet of the cell and the inner leaflet of the cell membrane. At this time, investigators studying the signaling of these GPI-anchored proteins noticed that whenever they isolated these GPI-anchored proteins in these Triton insoluble structures from cells in which signaling was occurring, they could find lipid-tethered inner leaflet components that were in fact associated with the GPI-anchored proteins. And these lipid-linked inner leaflet components were part of the Sark family tyrosine kinase complexes. IgE receptor, that is the immunoglobulin E receptor, was also found in detergent-resistant membranes, and detergent-resistant membranes were important for their signaling activity. The B-cell receptor was also important, was found in these detergent-resistant membranes, and these lipid domains that the B-cell receptor associated with was also thought to be important for the signaling. Putting all these things together, I think by the early to the mid-90s, there was an operational definition of rafts developing. On one hand, components that were associated with this detergent-resistant membrane were thought to be part of rafts, and on the other hand, if one was able to perturb the lipid constituents in the cell membrane, for example, cholesterol and sphingolipids, by using cholesterol chelating agents or metabolically depleting the levels of these lipids in cells, then an operational definition of a particular function in the cell could be associated with that function in the cell requiring the role of rafts or requiring rafts in that process. For example, sorting of membrane proteins, if they were associated with detergent-resistant membranes, and the sorting was perturbed by cholesterol and sphingolipid level perturbation, then it was thought that the sorting of those membrane components was occurring by these membrane rafts. So, in fact, this operational definition is a very simple experimental tool. It's easy to use, and a lot of people, a lot of investigators in the field wanted to know whether their favorite cellular process was involving rafts by these criteria or not. And from the mid-90s to I'd say, till today, a large number of researchers have used this criteria to make claims about membrane rafts in their favorite cellular process. But it also, I think this idea that components could be segregated in cell membranes has also driven a lot of research in the artificial membrane arena, where people have spent a lot of time trying to investigate the phase behavior of these detergent-resistant membranes. And we'll get to that in a little bit. The cellular context, again, of what these detergent-resistant membranes look like has also been investigated. And I think a lot of research now indicates that perhaps these simple operational tools may not be the best way to look for membrane rafts. But if we examine the literature on this detergent-resistant membrane composition, for example, it's a composition where you have equimolar cholesterol, sphingolipid, and phosphatidylcholine. If we examine this composition in a wide variety of systems, in giant unilamellar vesicles, in supported membranes, using fluorescently labeled lipid probes that could partition into different types of lipid phases, a whole wealth of literature has emerged. And this has been driven by the fact that this particular type of lipid composition supports two types of immiscible lipid phases. It supports two types of immiscible lipid phases in a fluid membrane, where one of them is a liquid ordered phase, and the other is a liquid disordered phase. And these two phases coexist in the membrane. The liquid ordered phase is defined by a phase where there is limited translational mobility, but there is no long-range order. Whereas a liquid disordered phase is a phase where there is there is no short-range order nor limited translational mobility. So these two phases coexist in cell membranes, and the liquid ordered phase is a phase that seems to correspond to a phase with composition of that of the detergent-resistant membrane. And soon enough, Debbie Brown and Irvin London in the early 90s, in fact, showed that there was a connection between liquid ordered domains and detergent-resistant membranes in artificial membranes, and made the link with what the detergent-resistant membranes might be revealing in a cellular context. So liquid ordered domains, which were correlated with detergent-resistant membranes, became in fact, a characteristic or a property of membrane rafts present in cell membranes. And if you look at the phase behavior of these raft composition artificial membranes, one can see sort of macroscopic segregation of lipid domains. In fact, you can see regions of the cell membrane which have, or regions of this artificial membrane which can concentrate GPI-anchored proteins that have been added to them, and in fact exclude phospholipids that have been put into the same membrane. So these artificial membranes, in fact, revealed the capacity of lipids to naturally segregate components in cell when they were introduced into the same membrane. The phase behavior of raft composition in giant unilamellar vesicles was another great push to understand cholesterol, sphingolipid, and phosphatidylcholine composition variations in these GUVs. And what people have found is that if you add cholesterol to these GUVs, you see a dramatic homogenization of these liquid ordered and liquid disordered membranes that could be present in conditions where cholesterol is completely absent. So if you have, for example, phosphatidylcholine, saturated acyl chain containing choline, and unsaturated acyl chain containing choline lipids, they segregate in the membrane. And then if you add cholesterol, those different domains seem to get mixed up. So it appears then that cholesterol is able to modulate the nature of these domains that are present in these artificial membranes. And the lesson that we have learned from these artificial membranes is that the role of cholesterol is sort of rather an ambiguous one, where at one level it's able to promote the formation of domains under certain conditions. At another level, it's able to mix domains. And this could be because the different kinds of sort of chemical interactions that cholesterol has with other lipids in the membrane. Cholesterol, in fact, is thought to interdigitate between the acyl chains of lipids and fill up spaces within lipids. So if lipids like unsaturated chain-containing lipids are present, cholesterol might go in and fill up spaces there. Alternatively, it could help in stabilizing the saturated chains of phospholipids and extending the chains out and making them more rigid in their structure. So cholesterol could have multiple roles, and each of these roles could be exhibited in different contexts and different times. Cholesterol could also form condensed complexes with specific types of lipids, and work from Harden-McConnell's group has shown that cholesterol and sphingolipids can form specific complexes in cell membranes, in artificial membranes, albeit these were monolayer experiments. Other lessons that we've learned from artificial membranes is that there could be a transbilayer coupling of lipids across the outer and inner leaflet. The charge of the lipid head group could also affect the partitioning of a lipid component in a liquid ordered domain with respect to that of a liquid disordered domain. But I think the question remains is, are these detergent-resistant membranes pre-existing structures or functional domains in cell membranes, or are they some artifact of the operational criteria that people have used to isolate them? I think a very important observation was made by Herklotz and Joachim Seelig, where they showed, using quantitative physicochemical studies, using isothermal calorimetry, that when you added Triton to artificial membranes that were homogeneous, you could in fact generate domains that were relatively ordered. And this was particularly true for membranes that contained sphingomyelin. And they argued that this was because Triton did not like to associate with sphingomyelins and somehow pushed sphingomyelins away while solubilizing other lipid components in the cell membrane. So in fact, they argued that the addition of the Triton or a detergent to the cell membrane could influence the organization of lipid components present in the absence of the addition of Triton. And in fact, in some cases, even generate ordered membrane domains when the Triton was added into these systems. So I think the detergent-resistant operational definition suffered a major setback by some of these very detailed observations made in artificial membrane studies. Another, I'd say, setback to the idea of membrane rafts in cell membranes was from experiments again by Michael Edidin, where he noticed that when you removed cholesterol from cell membranes, the cell membranes seem to suffer a disconnection from the underlying cytoskeleton. In fact, the cortical actin that was present beneath the cell membrane was disrupted when cholesterol was taken away from the cell membrane. In fact, these PIPs, or phosphorylated inositols, or phosphorylated phosphatidyl inositols, were selectively removed from the bilayer by means as yet not well understood when cholesterol was removed from cell membranes. Not only that, Antonella Viola and colleagues noticed that when you deplete cells of cholesterol, the intracellular stores of calcium were removed, and the plasma membrane of cells tended to be more depolarized. So these were artifacts that were associated with these experimental ways of removing cholesterol from cell membranes. So where does this leave us about in terms of what lipid rafts might be like? So are they creations of these operational definitions? And I would like to argue that this is in fact the case, that the lipid rafts, as observed by these different criteria of detergent resistance, of cholesterol perturbation, cholesterol level perturbation, are in fact in some sense an artifact of the operational definition itself. And then where does this leave us with in terms of the raft hypothesis? Here I think the cells are telling us a different story. The cells seem to be saying that there is a definite need and a reason to expect specialized membrane domains endowed with specific function, especially in terms of the sorting and segregation of components in the membrane to deliver components to different aspects of the cell surface, to engage with signaling across the bilayer of lipid-anchored components. In fact, all these observations suggest that there must be membrane domains which exist in cell membranes, and these operational definitions have merely been correlates of some kind, but have not really revealed the true nature of these membrane domains. So I think what is needed at this stage is to revisit this raft hypothesis. And I think the questions that we should be asking are, can we visualize specific regions of the cell membrane where lipids and protein components are concentrated? Can we literally look at these regions where components of certain functional content and context are concentrated? And can we observe them, and can we watch them functioning in cell membranes? And in fact, is the membrane really a patchwork-quilt where lipids are organized in some form or the other, perhaps with the help of proteins? And I'm now at the end of this part, where I'd like to make a break, and in parts 2 and 3 of this iBio seminar, I'm going to describe how studying the endocytosis of GPI-anchored proteins has in fact compelled me to re-examine the notion of membrane rafts in a living cell. And in the part 3, we explore what these ideas have revealed to us about membrane organization in a living cell. Thank you. Yeah.